Good afternoon, everyone present in this session of Faculty Development Program organized by Assam Downtown University. So, on behalf of Assam Downtown University, I, Mohidul Islam, welcome you all to this second session of the Faculty Development Program. In this session, the valuable speech will be delivered by Dr. Ovik Kumar Ghosh, who has 15 years of research and industrial experiences. He did BSc in chemistry from Jadavpur University, Kolkata. Then he completed his post-graduation from IIT Mumbai and has been awarded PhD in 2007 from Georgia Institute of Technology, USA. He worked as postdoctoral fellow and senior postdoctoral researcher in several universities and institutes in USA. He was associate scientific manager at Bicon Research Limited, Bangalore. He also worked as principal scientist in Biologic Division of Dr. Reddy's Laboratory, Hyderabad. Then he also worked as principal scientific manager at Biocon Biologics, Bangalore. And finally, he is now the director of pharmaceutical science project led at Algotech, Iceland, having the responsibilities for complete development of multiple biosimilar projects. So, I welcome to deliver the valuable speech by Dr. Ovik Kumar Ghosh. The stage is yours now. You can start this session with this valuable.
Thank you, Dr. Islam. Can you hear me?
Yes, sir, you are audible.
And you are able to see my screen?
Yes, sir.
Okay, thank you. Welcome, everyone. Probably I would be the only one here who doesn't have much teaching experience or just coming from the industry. And I just wanted to add also that I stayed in northern part of West Bengal for a long time, and I studied in Jenkins School till my secondary exam. So I'm quite fond of North Bengal, and I've been to Assam for many a time. So I'm quite fond of that area. And so coming back to the presentation, I just wanted to give a brief overview of an area that is being developed for last probably 10 to 15 years, and then it is expected to grow a lot in next 10-15 years. This is the biosimilar development that I want to discuss a little bit. It would be more of a basic understanding of biosimilar development, and then I would present a couple of case studies or papers to explain the procedure in more details, or explain more scientific angles associated with the biosimilar development. And I also want to emphasize that maybe industry work may look like something that may not involve that much of research and development or core scientific expertise, but these areas are quite niche areas, I would say, where a very sound scientific knowledge is important. And as I go through the presentation, you would see that how different areas of biological sciences, pharmaceutical sciences, and even organic chemistry and analytical sciences come together to build this area. Yeah, so without further ado, I'll just go to the next slide. Sorry, it's not moving. Sorry. Yeah. So, so just since I'm working in a public company and also worked in a public company previously, so I just wanted to provide a disclaimer that this presentation is only consists of general knowledge that I have gained in my 10 years tenure in different biosimilar industries, and it doesn't reveal any confidential information from my current or previous organizations. Okay, so now coming back to the agenda of the presentation. So I just want to briefly go about what is biologics, then what is biosimilar, then what is biosimilarity, and then and to go over very briefly to provide a basic understanding of how biosimilar development works. It's quite a vast field, but I'll try to provide a summary on what are the functions involved in this development. And in general, I have worked more in analytical sciences part, so obviously the presentation would focus more on analytical sciences, which is if not the one of the most important factor in this whole journey of biosimilar development. And lastly, I would provide some example from literatures and case studies how biosimilar development work, or how to show the biosimilarity. I've chosen one article from a group in China where they have shown a biosimilarity of a product, and then another article by me, which is a work done in my previous organization, where we have used several scientific analytical knowledge to actually troubleshoot an issue during a biosimilar development. Yeah, so we'll go into more details as we could go there. Since it is online collaboration, it's difficult to present when I'm not able to see anyone. But I'd say that if anyone have any question, it would be good if they can probably ask then and there. Probably it's okay if you want to interrupt me and ask. It is perfectly fine with me. It's always easy to explain when we are at there. Okay. And going back to the presentation now. So first topic is what is biologics. So the biologics is a drug that is it's a drug to start with. It's a product that is produced from living organism and contain components of living organism. So living organism is cells, and the components of living organisms can be protein sequences, say DNA, clone. These are the basic thing that makes a biologics, and it includes a wide range of products such as vaccines, blood and blood components, allergenics, cells, gene therapy, tissue, and recombinant therapeutic proteins. So the main focus of today's talk it would be the recombinant therapeutic proteins, because this is what actually nowadays as biologics as or biosimilar industries are targeting on because of their need, the unmet patient need, and also where their usability or utility in several diseases which are difficult to cure. And so, as we are more familiar with small molecule based drugs like these, so in contrast of the small molecule based products or drugs, these are so the small molecule based products which are like a chemically synthesized and their structures are known. So their basic structures are known, and it is uh, understood how to synthesize the molecule. In contrast, the biologics being a protein, and it's a very complex structures. So it's not a linear structure as for proteins. It has multiple structures, and it's very difficult to characterize because of its size, its complexity, its folding, and and association of these things with its function, right? So the main purpose of a drug is its function, right? So it needs to work properly, as well as it needs to be safe for the patient. So these are the two important things of a drug. And being a big protein molecule, it's very difficult to characterize everything of that product, and thus it takes a huge time to actually develop a biologics. So most of the biologics in market have been developed in almost like 7 to 10 years. So so you can imagine the amount of work that can go into it, and as well as the risk for a company where you are actually uh, spending money on research and development to make something for 7 and 10 years. So you're not gaining anything from that product for 7 to 10 years. Most of the time, when you're talking about the innovator biologics, we're talking about 10 years, right? So obviously, so because the complexity is the key here. However, as I mentioned, there is a very huge unmet need, and that actually leads to my next slide, where we more talk about the why the biologics are so important, why we are even talking about it, right? So if you see this figure here, this is kind of depicts the importance of the biologics. And sorry for the sound, just a second. Sorry, I'm back. So yeah, so going to the importance of the biologics. So as you see here, these are the drugs that the cells of the drugs worldwide in the last year, basically 2021. And as you see over here, the apart from the drugs that are vaccines directly indicating COVID-19, so these two are the ones. And so the COVID-19 vaccine from Pfizer and for Moderna. And as you see over here, the other drugs except this Eliquis and this Revlimid, these two are small molecules, as well as Imbruvicia. Other than that, the Humira, Keytruda, Eylea, Stelara, all are biologics and recombinant proteins biologics. Okay, and actually, because of the COVID, these have been changed a lot in last year. But in general, Humira has been the most priced and the most marketed drug in for last few years. So this actually shows the importance of the biologics in the medicine world. And as you see over here, so obviously because of the time required, the money spent on actually making these drugs, the discovery, the manufacturing, the clinical trials, and everything makes these drugs very expensive. So obviously, all the companies who are making the drugs are motivated towards meeting the need for the patients. They also have to make profits to move ahead, right? If they're not making profit, they cannot make drugs. Then there is nothing. I mean, they cannot supply the drugs for the patient. So obviously, because of this huge money spent in biologics development, also although these drugs may actually prolong your life in some cases and save your lives, they are very costly. For example, Humira is a drug for rheumatoid arthritis, and this probably needs to be taken lifelong once you have it. So it's basically helping you to manage the arthritis, right? So it needs to be taken weekly, and a cost of Humira in say for a 2 weeks dose is, even after all insurance claim and everything, I'm talking about US dollars in US, it's around 6,000 US dollars. And if you want to convert it in rupees for countries in India or China, so that would be even huge, right? So obviously, it's not possible for everyone to afford these biologics, although these are available in market, right? So that's where the comes the need of biosimilars. So once these biologics are going off patent, the generally after 10 or 12 years, depending on which region we are talking about, there is a scope of biosimilars to come into the market, which can be developed in a lower price, because here we are not really innovating the molecule, because we know already what molecule to make, but we are making it in a different way, but as good as the biologics that is in the market. So obviously the development cost is less. Maybe instead of 10 years, it might take 4 years, 5 years. And that obviously helps us with the price. So most of the biosimilars in the market right now are more or less almost 50 to 40, even maybe 30% less costly. I mean, 30% cost. So basically 50 to 70% less costly than the originator biologics that in the market. Okay. So considering all this, uh, so the use of biosimilar or the need of biosimilar has become a very important factor in the industry now. So now, what is biosimilar? Sorry, I have to move it in here. What is biosimilar? So biosimilar is a biological product that is highly similar to the existing reference product, which is the existing biologics that is in the market, and has no clinical meaningful differences with that existing product. So what does it mean? So as I told you before, these biologics are very complex molecule. So these have very complex structure. So it may not always be possible to actually mimic the exact biologics, right? Because since there are structural differences and complexity around the structure, you cannot always mimic the structure. So there would be differences all the time. However, these differences should not impart any clinically meaningful difference. Okay. So basically, what I mean is that, as you see in this picture over here, so these say, if you see, this is a reference product and this is a biosimilar product. Okay. So as you see, there, other than the protein backbone itself, there can be other things attached to it, like carbohydrate chains and many other things. And as you see, there might be small differences in terms of those, right? And as long as these differences does not impart any impact in the clinical trials, say like especially safety and efficacy of the product, this can be considered as a biosimilar, approved biosimilar product of the reference product. So for all the regulatory authorities like FDA, EMA, or even for our Indian regulators and all other local regulators, there are guidelines in terms of uh, how close or how similar this products needs to be to the reference product, and what are the clinical trials that needs to be done to show the differences are not meaningful and all. So generally these things are, although it's developing, I would say, for developing authorities, right, like FDA, EMA, or PMDA, Japan, these are more fixed or certain in terms of these, while the other authorities like smaller European authorities or India, China, these are still, I'd say, still developing in this stage. So these guidelines are still being developed and modified over the time to make sure that, so the all the regulators always want these molecules to be in the market. Obviously, it helps them to run the healthcare, right? Because these would make uh, life-saving drugs available in the market with which would be accessible for most of the people, right? So obviously, all the regulations are around making these drugs available in the market, but obviously, the strictness around the safety and efficacy of the products are always in place. And so, and one more difference that I wanted to point out is between a biosimilar and a generic, right? For a generic product, you already know the structure of the product that you're trying to mimic. Same as a biosimilar, you already know what biologic you want to mimic. You know it's a protein. You know the amino acid sequence of the protein, so you know what you want to make. However, the difference being, as I mentioned, for chemically synthesized generic product, it it has to be exactly similar as the exactly same as the originator drug, right? Because it's a defined molecule. You cannot have a derivative of a product as a generic, right? Because it will change the product. It may change the functionality of the product totally, right? So that's where the differences are there between the biologics and between the biosimilar and the generic product. Okay. And as I mentioned that when you are applying for a license for marketing of a biosimilar, the manufacturer needs to demonstrate that there is no clinically meaningful difference between the reference product, which is the biologics that is in the market, and the biologics product in terms of safety, purity, and the potency. Potency is efficacy that I'm talking about. So these are generally demonstrated by the PKPT studies and a clinical immunology or additional clinical studies. Right now, although the clinical studies are required at the end to definitively show the safety and efficacy of a product, the research and development part and the part where we are actually using our general scientific knowledge to actually make the product and test the product in the lab is also very important. And as you see here, so as I was mentioning over the time, so this is the term called biosimilarity, where you actually show that your proposed biosimilar product is a biosimilar to the reference product. So that is called, you establish the biosimilarity. So that is the goal of the development of a biosimilar product, that you have to do all the things to show the biosimilarity of the product to the reference biologics and then present it to the regulators to get the approval. So now, as you see in the right hand side, I show you a comparison of the approval pathway of a reference medicine development, which is a biologics product, innovator biologics product, versus a biosimilar development. So, so these two are, so this is an FDA terminology in terms of which regulatory pathway. So basically 350A, 351A is the pathway for approval of innovator biologics product, where 351K is approval of a biosimilar product. Okay, so this is just for information. So main focus is in here. So as you as you see in here on the left hand side, the for a innovator product and a biologics product, the clinical studies are the main foundation. So basically, since this is obviously some analytical characterization of the drug is needed to understand the clinical outcomes better, but it all depends on the clinical outcome. Okay, so when you are trying to make a innovator product, you will probably have multiple candidates of a product that you will first test in a clinical phase. Then based on that, you actually would try to figure out which is actually working best in a clinical scenario, and then you want to go into more details of that, and you want to do more development around that. Okay, so that makes the clinical and also, since it is a new biologics product, your clinical trial sample size also needs to be much bigger, right? Because there is no precedence of that product in the market. So you have to have a much bigger clinical study and much longer clinical studies, and your most of the emphasis of your development is on the clinical studies. However, for a biosimilar development, it's the other way around. Okay, so here you are making a molecule that is already in the market. I'm using the word molecule because it's the same molecule as the reference biologics, right? So the clinical effectivity of that molecule is already established. So this molecule is in the market for 10 or 12 years. So obviously, the regulators already know that this molecule is clinically fine. So what you need to show is whatever product you have made, whatever molecule you have made, is clinically similarly effective, right? So here your that is why it's called a confirmatory clinical phase, right? The main focus here is in the lab work, where the analytical sciences, where you have to show using different state of the art techniques, actually to show that the product that you have made is analytically similar to the biologics product. So once you have showed this part, this base part basically. Then you can apply for the clinical studies, which would be much smaller studies, and because the clinical efficacy of the molecule is already established, and you have already established the similarity through analytical studies. So these would be much smaller studies, and the base of a biosimilar development is showing the analytically similar part. Okay, so obviously that analytical work that is involved in establishing this analytical similarity or biosimilarity here is very important. Okay, so now what I'll do is I'll slowly sorry go over the different scientific functions that are involved in biosimilar development, as well as how the flow goes. Okay, so basically it starts from reference biologics, that is the innovator biologics product that is already in the market, which you want to make the similar product. So understanding that product makes is very important here, right? Because you want to make the same product, so you need to understand that product very well. And as I mentioned, these are complex proteins, so it's very straightforward to understand all the scientific features, the physical, chemical, and functional properties of the reference probiologics. So this needs quite an advanced technology to understand this part. So this is the first beginning part of the and a biosimilar development, to understand the reference biologics. Obviously, you need to know the molecule so you want to make. So generally that for a company it is more of a business decision, like which product to target, and that can be a decision based on the which therapeutic area that company is focusing on, or it can be a market intelligence call, like after, say, from 10 years from now, which product can be the most important product. So there are several ways to define that, but it's more of a company decision to decide which product to make. And once that is decided, the first part is to actually buy those product from the market for a small amount for research work, obviously, and then understand the physical, chemical, and functional properties in details, right? What is the structure of the product, what are the impurities are there in the product, what are the amount of the impurities that are there in the product, how the functional properties work, what is the mechanism of action of this product, how in a lab, in an in vitro scenario, you can demonstrate the mechanism of action of this product. So this is a quite huge work that goes into that. Obviously, being because of the reference biologics is already in the market, there might be some literature available on these, but most of the things are not that much available, and um, you need to actually make them from the scratch. And also another important thing here is the intellectual property, IP. So many of the, in general, when our innovator biologics get accepted for marketing for FDA and EMA, 10 years in FDA and 12 years in EMA, that the exclusivity exists. So after that, it becomes open, so anyone can market a biosimilar product. However, innovators would have patents around the formulation of the product, or some of the processes that are involved in the manufacturing of the product. So it is very important that the biosimilar product company knows about those patents and try to avoid the conflicts, right? Because if they're trying to mitigate a patent or conflict in a patent that actually ends up being a long litigation process, and at the end of the day it gets delayed. So in short, the first part is knowing the reference biologics and all the patents around it very thoroughly. So next comes the actual development of the biosimilar. So you know now the what is the product you want to make, you know the amino acid sequence, and most of the time it is available in which host cell line this product has been manufactured. So when a biosimilar company starts making the development, so the first part is the cell line development, where they select the host cell line and the right clone, right? So here, we are basically making a recombinant protein. So you are genetically engineering the clone. So you're incorporating the appropriate cDNAs and making the clone, and then trying to generate the protein in the host cell line, right? So there can be multiple clones. So there is a process where you actually select the clone. I'll come to it little later. And so basically here your goal is, there are two goals, right? One is obviously you have to attain the biosimilarity, right? So that is the main goal here. And also the output, right? So you want to make a biologics that you want it to reach to the patients, right? So if you are not able to make it enough, you won't be able to do that. So obviously the output is very important. So the yield, how much you are making, so that is very important. As you see in the other steps also, this is used as a key factor to determine or select the clone or the development pathways. Okay, so once you have selected the host cell line, and then you have selected the right clone, or at least whatever you think as the right clone, you have selected a clone to go ahead with. Next is the cell line development. So here also, sorry, the cell culture part, the upstream development. So here also, so basically now you are generating protein from a clone in a host cell line, right? So there are many things that are involved in the cell culture process that can be modified, optimized to make sure that you can you can make a product that is biosimilar, and as well as the again the yield part is very important. You can make sure that you are making a good amount of the product, right? Next comes the downstream development. Okay, so in the downstream development part, it's the most important part is the purification part. So basically this is the purifying the protein. So basically you have now make a protein from the cell culture, you have harvested the protein, but that protein doesn't come out from the cell culture as only a protein, right? It comes with other host cell proteins or cell DNAs. It comes with impurities. It may have some other molecule in it, although very small amount, but can have. So obviously, and it can be not, it can be something that is not exactly biosimilar, right? There might be some differences still there, so which can be taken care during the purification process, which we call the downstream development. So these are mostly the chromatography processes, and at the end involves some filtration processes also. So here also the goal is to get rid of the impurities as much as possible, right? Because these can be issue for the safety of the product. Like if you have the host cell proteins which are coming from maybe a CHO cell line or murine cell line, SPTO cell line, so which can be bad for your your system. So definitely, and these clones, they come out can come up with other viruses and everything. So these all needs to get purified. So this part is taken care in during the downstream development. So after the downstream development, what we generally get is called as a drug substance. This is the active product which is purified and everything. Now you have to make sure that product acts properly in your body, and also when it is stored, it can be stored for a longer time, at least the time that is the reference biologics can be stored, right? So that is done by development of a formulation, right? So you have the active substance, it needs to go in a formulation, right? So that can be a buffer most of the time, with some excipients or stabilizers to make sure the protein is stable. And also this part also involved the selection of a container, say like where your biosimilar product would be. It's in a vial, or it's a pre-filled syringe, or it's an auto injector, and all those development happens in the formulation development or drug product development part. And what goes hand in hand with all these steps is the analytical development. So as I mentioned before, so you when you want to understand how your reference biologics, you need analytical techniques to understand the physical chemical properties, understand how pure it is, understand how functionally active it is. So from the beginning to the end, or the each step, your whatever development these process functions are making, the outcome is it has to be biosimilar. So basically, they need to check it by the analytical methods or analytical techniques to make sure that they are in the right track, right? So the analytical development, although the box is I put it in a smaller box, it actually expands from the beginning of the biosimilar development towards the end. Okay. And so this analytical development generally involves as development of analytical methods which are appropriate for understanding the quality attributes of a reference biologics and the biosimilar. Next slide, I'll discuss more in terms of what is quality attribute mean. And then also one important part here is even for process development also, when we it needs to run in a repetitive manner, right? There can be small variations that cannot be avoided, but all the time this process is run, the cell line development, the cell line upstream, downstream formulation, it needs to yield the same product, right? So and the it's all the time these functions needs to act as a same way, it cannot be different. Similarly for analytical methods also, they cannot vary. It's not a one time analysis, you have to analyze the batches of the biosimilar product over a long time, when I mean over the lifetime of the product, right? So these methods should be very robust. So that's why the method development and the qualification part takes care of the robustness as well. So it is actually a demonstration that the method is able to estimate or measure some quality attribute repetitively in a same accurate manner. Okay. So basically that's what the goal of the analytical development part is, to develop these methods and qualify these methods, and this analyze the reference biologics and propose biosimilar product. Okay. So this is in a nutshell of what are the different functions involved in biosimilar development. Then I'll go one by one, in little details in each of the functions. Sorry. Yeah. So first part, as I mentioned, is the understanding on the reference biologics. Okay. So here I have shown a monoclonal antibody, which I mean most of the marketed biologics and biosimilars are monoclonal antibodies, IgG1. There are a few IgG2 and 4 that are coming into market, but mostly it is IgG1 and IgG1 MAPs, monoclonal antibodies. Right, so it have a Fab region which actually binds to the target and the Fc region, and there some of the so the Fab region contain that complementary determined region or CDRs that binds to the antigen, and then there are for FC region there might be some functionality of the FC region as well for some of the product. I'll show one example during the case studies today. And also one other important thing, there are generally with maps there is sorry just pointer. Yeah, generally with MABs, there is glycosylation. So basically, a carbohydrate structure that gets attached to the MAB around in the FC region, which is also sometimes important for its function. Okay. So basically, then how do we understand this biologic? So I have shown very this is a very simple depiction, if you say. So this general ideas are primary structure, which is the amino acid sequence, and higher order structure, which consists, as you see there is some disulfide link, which actually holds the two heavy chains of the product, as well as the heavy chain and the light chains over here. These all are the disulfide links. So characterizing this disulfide bridges, then knowing the secondary structure and the tertiary structures, then the PTMs, the post-translational modifications, something like the glycan, the glycosylation that I just talked about. Then this is a protein that is going through so much of stress, I would say, when it is being produced and then purified, and also it may get oxidized or deamidated. There are methionines or tryptophans that can get oxidized, or asparagines that can get deamidated. Then there can be product related impurities like aggregates. So this is a single map, right? It can multiple map can come together and form aggregates. So that obviously the function of an aggregate is different than a single product, right? So you don't want much of aggregate in your product. So that's why it is a product related impurities, right? Then there are fragmentation. So this product can break into smaller products while making, as the production itself is a process. It's a stressful process, right? So this is the fragmentation. So that can also happen. So that is also an impurity. Okay. Then the last but not the least, the biological activity. So where you need to understand the target binding. So it's a function of this map is to bind to the target, right? So here you understand the target binding, and then if there is any FC functionality. So again, this is a very what should I say, very simplistic way of showing it. Obviously there can be different complexities around these, and your product may not be just a simple IgG1 map. There can be other, it can be a fusion protein, and also which would bring in other complexities and all. But this is in a nutshell, as a basic understanding. Okay. Now I'll just touch upon. Sorry. Oops. Sorry. Yeah, some analytical techniques that are generally used in the analysis of reference biologics, as well as the actual product. So as you see here, so main analytical techniques that are used in biosimilar industries are based on mass spectrometry, liquid chromatography based separation, and spectroscopy and electrophoresis based separation techniques. So basically the primary structure is mostly coming from the mass spectrometry, where the intact mass, so these are very big proteins. We're talking about 150 kilodalton protein. So it needs high resolution mass spectrometry to analyze the intact mass, and the subunit mass means the mass of each of the light chain and heavy chain, which are also 50 and 75 kd big. And then the peptide map, where you actually digest the protein with different enzymes to get the individual peptides, and by LCMS or MSMS, you actually depict the sequence of the protein to make sure that the protein that you have made is exactly at least the amino acid sequence should be exactly the same as your target biologics, right? Then for higher order structures, as I mentioned, disulfide is one analysis that is done by LCMS. Then the higher order structure as a secondary or tertiary structures can be done by CD. Then thermal stability, it's also part of structural integrity of a product, is done by differential scanning calorimetry. Then more advanced techniques can be sorry, can be used to understand local structures better. So as you see, these methods or these techniques like a CD or DSC, these are more gives an idea on a structure in a more global level, like how much of alpha helix is there, how much is beta sheet is there. So it's more of a global understanding of the structure, while very small differences, the local structural differences can be depicted by more high-end technologies like hydrogen deuterium exchange, MS, or even NMR. And then if I go into the product related impurities like aggregates and fragments, these are aggregates are mostly by size exclusion chromatography, where the fragments are, I mean, SDS page has been used, but most of the industry has now moved more capillary electrophoresis based techniques, CESDS. And for post-translation modifications like glycosylation that I mentioned is a very important part, which is generally done by UPLC or HPLC. Then oxidation, deamidation, these all can be done by UPLC, HPLC, and sometimes it can be clubbed with mass spec as well to get more detailed information. And then charge variants, these are generally done by the ion exchange chromatography and or capillary electrophoresis based techniques. And then biological function, generally it is, one is the efficacy of the potency of the product, that generally is shown by a potency assay, okay, which is basically a cell-based technique. And then ELISA or surface plasmon resonance based techniques that generally shows the binding part, where the target binding ability of the protein is measured. Okay, so these are I mean, in general, so these are the techniques that are generally used to analyze the reference biologics, to understand the reference biologics, and then these techniques are eventually used over the development of the biosimilar product, and then analyzing the biosimilar product at the end to show the biosimilarity. Okay, if we go to the next slide. So so basically the biosimilar development, so as I mentioned, you are analyzing the reference biologics first, right, to understand it. So what you get out of it, right? So you get out of it is the target, right? So what you want to make. So how the target is defined by numbers. So it defined by the numbers, it's like how much aggregate you can have, right? You analyze many innovator reference biologics product. You see that so these products have say 0.2% aggregate, 0.3% aggregate in it. So now you have set your target, right? So you have to make a product which also has 0.2%, 0.3% aggregate, right? So this is how it works. And now it might be that you are not able to make a product which has 0.2%, 0.3% aggregate. You are making a product which has 0.5% aggregate. Now, is it all gone? No. So always remember, in the biosimilar, this term is, it needs to be, the difference can be there, but it cannot be clinically meaningful. So as long as by biological experiment you can show that these small differences that you are seeing, obviously if you have if your target is to make 0.3% and you are making 30%, that is obviously a no-go. But you are also in the very, in a very small a very close region, say instead of 0.3 you are making 0.5. So these type of differences can be explained by appropriate biological testing. Okay, I'll come to that, how does it work. So basically your first part is to actually set the target, right? So for that, first you understand what are the quality attributes I want to target, so that these are called the critical quality attributes, right? So these are the quality attributes that are directly related to the safety and efficacy of the product. Okay, so generally when you start a development, you would have a literature available, like open source articles, regulatory documents available on the reference biologics. Also, you can have literature available on similar molecules like IgG1. Say you are making a biosimilar which is a IgG1, but there would be there might be information available on other IgG1 products. So you can use that information, because most of the time, other than the exact binding CDR or the functions, most of the time the general properties may remain the same. Okay, so those information can be used judiciously, I would say. So that is the first part, to understand the critical quality attributes. Then set up a statistical approach. So basically you will analyze many reference biologics batches, right? And as I mentioned, these are very complex product. Even the innovator will not able to hit the same thing all the time, right? Say we are talking about, say, aggregate. Say in one batch of innovator product it might be 0.2%, in one batch it is 0.3%, in one batch it is 0.4%. So what should I target, right? So then we have some statistical approach we analyze many reference biologics batches, and generally it's at least 10, to get a range, right? So that is range that is there in the innovator biologics product. And now we can do some statistical analysis on that range to say that, okay, so I have a range, and now I'll do some statistical approach, say mean plus minus 3 SD, to have a target range for my product. Okay, so that is the statistical approach you can define. And also for some quality attributes, there is no way you can have a quantitative approach. So there can be qualitative approach can be taken. For example, something like a cyclodichromosome CD. So there is not much quantitative approach you can take. It's just two different profiles that you have to match. So these are the qualitative approach. So basically, it's very important to understand the critical quality attributes and the statistical approach you want to take for each of the critical quality attribute, if it is a quantitative attribute, and if it is a qualitative attribute, how are you going to compare? Then comes the analysis, where when you analyze multiple batches or lots of reference biologics product using state of the techniques, and you try to analyze batches from different ages. Okay, so that also the effect of aging of this product also get into account. And based on that, you apply that statistical approach to obtain the quality range. So this is basically the target range of a quality attribute that you want to target during the biosimilar development. Okay, so now I move to, so once this quality, so this is the what we generally say is a homework phase, right, where you have analyzed the reference product, and then you have finalized this at the target ranges that we are going to work with. Okay, now I'll go by each of the functions. So as I mentioned, as analytical development is the most important function, because it is involved in throughout the development. So basically, once you have set the CQS, You then select the analytical techniques that can measure the CQA. So basically, your critical quality attribute CQSA is one of the CQSA is aggregate. So you select size exclusion chromatography to measure that aggregate. Now, you develop a method that is appropriate to analyze the reference biologics and biosimilar product. Say, for example, size exclusion chromatography. Your goal is to separate the aggregate and the main product in the liquid chromatography, right? So you develop your method such a way these two things, the aggregate and the main product, which is not an aggregate, are appropriately separated so that they can be quantified reliably, right? So that is the method development part. Now the method qualification or validation part, that is basically where you establish that your method is capable. So basically, as once you have showed that your method can separate your aggregate and main product, now you have to show it can do it repetitively all the time we are doing it. It doesn't matter which analyst is testing. It doesn't matter which instrument we are using. It may doesn't matter the buffer component, the vendor of the buffer component. So all these permutation combination needs to be done and shown that this obviously we cannot do it for a year. So and probably within a month you have to do all the permutation combination, different instrument, different analysts, different labs if possible, and all that, to show that this method is capable of accurately measuring that aggregate, for our example, over the time. Okay, in a repetitive manner. Then comes the actual analysis part. So once your methods are set, then you analyze the reference biologics, and then you analyze the biosimilar products as well during the development of the biosimilar, as well as at the end when your final product is ready to establish the biosimilarity. Okay. So if I then next function I'm going to discuss is the clone development. So this is the first part once you start your biosimilar development, right? So this part is basically you have multiple clones of the product in your hand, and you have to select the right clone. So first part is the selection of the host cell line. So generally it is an easy task because most of the times the reference biologic cell lines are known, and you generally use the same cell line to avoid any conflict or further scrutiny. But there might be situations where it is not possible, say for some products in the market where the innovator has used a murine cell line, which doesn't yield much product, right? Now, some biosimilar company may think that, okay, I would not use a murine cell line, I'll use a Chinese hamster ovary cell line to improve the yield of the product. Okay. So in that case, they have to probably show a more elaborate analytical similarity, or do more additional clinical studies to show that this switch of the cell line did not impact the efficacy or safety of the product. But there are examples where companies have done it. Okay. But general practice is they use the same cell line as the reference biologic. Then you start with multiple clones, like say, depending on how you are doing it, what is your technology, or you are buying it for other vendors, it might be 20 to 50 clones you start with. And the first screening is generally based on the yield. So which clone is yielding the best amount of product. So these are very small scale runs you do where you just check the yield, and based on that, probably you select 5 to 10 clones from the second screening. Now, the second screening, you already know the critical quality attribute. You already know the ranges that you want to work around, so that you then you try to see that which of the clone is giving you the closest to that range. Okay, so based on that, you probably select again 2-3 clones. Okay, so now the final selection where you check the generation stability of the clones as well, because you need to make the product for say next 20 years, 30 years, right? So that clone needs to be the each generation the clone needs to be stable. It should not change its properties, right? So that generation stability obviously you would not do for 30 years, but you can go through many generations and see that the stability is there, and then the yield and the quality attributes. And also this clone, this needs to be taken to the next steps and make a product, right? So that development needs to be done. So based on all these factors you come up with the final clone. Okay, so basically the clone development or cell line development in a biosimilar industry, so they select the host cell line, then they select the clone. So then once the clone is selected, now you have to make the protein from that clone in a cell culture environment, right? So that is the upstream development or cell culture development. So here basically you use different cell culture conditions in very small scale. So these are like amber bioreactors which can uh, make like very small culture, 250 ml cultures they can do. So this you can test different conditions like media condition, feed, temperature, pH, duration of the cell culture, etc., in very small scale to figure out which is the best for the clone and the cell line that you are using, right? Then you go to a little also you analyze the cell culture profiles like the viability, titer, metabolic profile, and the product CQS at the different conditions, right? Now, based on these results, you finalize the cell culture condition. So, which is giving you the best cell culture profile, the best yield, or the product quality, and that can be done in a bigger scale as well, right? Because this is all we are talking about doing things in. So, this is like 250 ml scale. This is probably a little bigger scale, 1 liter flask, and then you can check it in a lab scale bioreactor which may be 10 liter or so, 2 liter or 10 liters. But the actual production which will happen is in a huge scale, is a 2000 liter or 1000 liter. So basically you need to make sure that this process that you're designing here is scalable. So that can be done based on the understanding of the process, but that is definitely one thing that you need to look into when you are finalizing the process. So generally once the process is developed in a lab scale, it is confirmed in a higher scale, maybe a 250 liter or a 50 liter bioreactor. Once it is confirmed that this multiple times, say you make 3 batches uh, under those scales, and finalize that okay, so this process is stable, then we say that we can call it a process log. So you say that okay, the upstream, the cell culture conditions are finalized. So once the cell culture conditions are finalized, the next step is the downstream processing, that's the purification. Okay, so here the first part is to understand the what are the impurities that needs to be purified, right? That's where the understanding of the process and the product related impurities that needs to be cleared or controlled. There might be some impurities that you cannot make it 0%, say for aggregate, as I was talking about. You cannot make a 0% aggregate in the product. It might be 0.2%, 0.3%, 0.5%, but it needs to be controlled. It cannot be like 30%. So basically that way you need to understand what are the quality attributes that you need to control, and based on that you design the purification steps. So generally downstream processing involved multiple purification steps, and then the individual steps can be optimized, like a resin or the buffer, or the mode, the way that has been used, like a flow through mode or bind or dilute mode, those things can be optimized. Then the whole purification steps, so basically one after another, is performed to confirm the capability of the process. So here I've shown typical downstream processing processes, which goes through on protein chromatography, then viral inactivation, then there can be ion exchange chromatographies and an exchange cation exchange to further purify the products, then the filtration. So these all steps are executed to make sure that all the impurities are out of the product. Okay. Or control. I mean, wherever you cannot make it zero, it is very lower level. So after that, so the outcome after the filtration is the drug substance, as I mentioned, is the active product. That then the next step is the formulation part. And so in the formulation part, so you need to select the formulation components. So basically, these are the buffer components and the pH of the buffer and the excipient or the stabilizers that you need to decide on. So generally, again, if it is same as the reference biologics, it is always easy to, or relatively easier to show the comparability. But many of the time, these compositions are patented. So the biosimilar organization has to come up with a different formulation, which also should make the product stable. Okay. Then also the primary packaging, like it would be in a vial or a syringe. The selection of those is also very important. Like who would be the maker of the vial or the syringes? There are different sizes of vials and the syringes. Which one we need to take, and all these things needs to be taken care of. Then once these are you can then the stability studies generally are done to see how the product, if the product is stable under these formulations or different primary packagings, and based on those studies, the final formulation and the filtering process and the primary container is selected. So this is basically in a development scenario how you make a biosimilar product starting from the cell culture to the drug product. I'm not going into details of the next step, which is the manufacturing and regulatory approval and all this, but just to show. So once the biosimilar is developed in the lab, right? Then the process is transferred to a GMP facility, which is a good manufacturing practice facility, where it is actually manufactured in a much higher scale, because obviously we need to do it on a higher scale to supply the needs. Then the clinical trial, for the clinical trial, you do a clinical trial application, and then once the clinical trial application is approved, then you do the clinical trials. These are generally safety and efficacy part. So these now I'm talking about, if you remember that pyramid, I'm talking about the just a second. Sorry. Yeah. So I'm just talking about now the tip of the pyramid, where the clinical trial is done, and then based on the clinical trial result and the biosimilarity result, the biological license application is filled, and based on to the regulatory authorities, if it is FDA or EMA or Indian authorities or any other countries, Japan, wherever the company wants to market it. And based on that, if the approval is done, so generally the approval process involves quite thorough scrutiny of all the documents, process, how it is being done, and even actual inspections in the site. And based on that, if everything is fine, the approval and the launch is done. Okay. Okay, so this is basically my talk regarding the biosimilar development part. Um, I can go to the next part, which are the case studies. I will skip the first one because we have 45 minutes and last 15 minutes I want to keep for question answer. So probably I'll just skip the first case study part and I'll go to the second case study and okay. So sorry.
Practical, Department of Pharmaceutical Practical Final Exam, Diploma in Pharmacy, first year. No?
Sorry.
फर्स्ट ईयर हो गया उसके बाद है सब्जेक्ट कोड फार्मास्यूटिकल
Or maybe sorry. Okay, we'll go to the case study one first, and then if time is there, we'll go to the second because otherwise it would be difficult to understand the. Okay, so as I mentioned throughout my presentation, so the establishing the biosimilarity, the analytical similarity is a key factor, right, in the biosimilar development. So after all the development is done, you have the product, you are making the product multiple times, and then in your manufacturing facility. But before getting the approval, you need to show that your product is similar, right? Analytically similar to the reference product. So here, so there are guidance, of course, around in different, say for FDA, for EMA, or even for Chinese authorities or the PMDA, how to do that. And generally, this is done. This is done going through the appropriate guidance, and then also these are generally while it is being done, generally you can go for advice to these authorities and show these data and see if we are on the right track or not. Okay, so here in this paper, so the analytical similarity of a biosimilar candidate, it's called HLX01, is shown. So this is the MabThera is the reference biologic. So it's a product from Roche. It's generally used for the large B cell non-Hodgkin's lymphoma. So it's kind of a blood cancer, and then several other chronic lymphocytic leukemia as well as RA. It is used. This was
यहाँ पे टोटल 80 है हाँ और जो 20
Sorry to interrupt. Apurva Kumari, kindly mute yourself.
दे देना है ना हम सबको 17 18 3 नंबर देना है
yeah, so then so this was approved by EMA, European authorities in 1997 and FDA in 1998. So basically this is off patent for some time now, and there are many biosimilars available for this product in India. Dr. Reddy's is there, and then many other companies have biosimilar product in this. So so basically this so basically this paper talks about how the analytical similarity between this HLX01 and the MabThera is shown for this product. Okay.
कुछ ये मैं बोल रहा है रहेगा एक दो टॉपिक अगर ऐसा रहेगा तो हम
the biosimilar development pathway that I have discussed. Okay, so going to the next slide. Sorry. Yeah. So here the important part, if you remember correctly, in the approach I had said, the first part is to understand the reference biologics, right? To actually figure out your approach, right? So here, so their approach also, one important part is to know that which market you want to get the authorization. So this is a product that is produced in China. So they also obviously wanted to get approval in Chinese market as well as European market. Okay. So that's why they have shown the similarity here to the rituximab. So this rituximab is the MabThera, or rituximab is the name of the molecule, and MabThera is the name of the product. So they show the analytical similarity of this biosimilar product HLX01 with these two reference biologics. So rituximab from China, rituximab from Europe. So this is a common practice where different wherever you are filing, if you are filing in Japan or you are filing in FDA, when you are filing in India, the regulatory authorities want to see the biosimilarity against the innovator biologics marketed in the same country. Okay, so whenever a biosimilar company wants to market something in India, they need to show the biosimilarity against the reference biologics marketed in India. Okay, so similarly here their targeted was China and Europe, so that's what they have used. So they have used different analytical techniques, as I mentioned. You'll see some as the techniques that I mentioned previously, how the output looks like and how they're used in showing the analytical similarity. And obviously, as I mentioned, generally guidelines are available, EMA, FDA, and ICH guidelines that are used. And also, as I mentioned, that you need to use quite a number of batches to have a good statistical analysis, right? So here they have used 12 batches of this biosimilar product, 15 batches of this rituximab China, and 7 batches of this EU rituximab to demonstrate the similarity. Okay. Now coming to the statistical approach. So as I mentioned, the important part is to know the critical quality attributes. So they have actually understood the quality attributes and then ranked them. Okay, so based on, say, some are tier 1, some are tier 2, and some are tier 3. So for the tier 1, they have used a confidence interval approach. And then tier 2, they have used a mean minus mean plus 3SD approach. And the tier 3, these are the lowest risk. They're evaluated by the visual comparison. Okay. And also important part is to know the mechanism of action, right? So how the rituximab works, right? So as I mentioned before, generally how a mab work is it binds to the target through its fab, right? So generally the fab function is very important. This, as you see over here, the rituximab is binding to the B cell CD20 by the fab region, right? But for some products, as I mentioned, the FC function is also important, the lower part, right? And rituximab is one of that kind of example, where after binding to the CD20 through the fab region, the interaction of the FC part with the effector functions are also important. So there is a ADCC antibody-dependent function that is involved here, and CDC, which is a complementary mediator cytotoxicity function. So these two functions, which are actually the functions of the FC part of a mab, is important. So, and then there is this direct lysis pathway where only the fab part is important. So basically, if you look as a whole, so there are 3 mechanistic pathways by which rituximab works, and all of them would require the fab binding to the target, which is CD20, but 2 of them would also require FC functionality, the ADCC and CDC. Okay, so for this product, if when we are doing the characterization, when we are doing the biosimilarity, analytical similarity, showing only the Fab uh, functionality will not suffice. So you also have to show your FC part is also functional. Okay. So now I'll go one by one how the analytical similarity in this case is shown. Sorry. So this is the critical quality attribute. So basically, as you see over here, so these are different characteristics of the product, and they have based on the understanding of the literature and maybe some lab experience, they have ranked them. So they basically they either tier 1, tier 2, or tier 3, as I was mentioning before. So and their statistical approach is based on the tier. So tier 1 means it is very important. So basically it's very high risk if you are not matching. So that's why they have used the confidence interval as a statistical approach. Tier 2 is not very high, but still important, where they have used mean plus minus 3SD approach. And tier 3 is generally where it is not very important or cannot be measured quantitatively, where qualitative basically approach is used. Okay, so this is just a list of the uh, characteristic or the quality attributes, as you see, product quality attributes here. They have tested here, and these are the different analytical methods that I have already mentioned that are used to test these attributes. And as you see over here, there are a lot of FC functionalities, FcRn binding, FcR1 binding. So FC binding to its effectors are involved here. This is because, as I mentioned, it's the function of rituximab is not only dependent on its Fab activity, it is also dependent on its FC activity. So showing that FC activity is also intact and it is similar to the reference biologics is very important. Okay. Now I'll go into the results one by one to show how they show this each. As you see here, already the assessment is done. And there is, if you see over here, in most of the case, it is similar, identical, but are some cases, there are some differences. Okay, so as you see here in N-glycan, you see percent galactose, percent fucosylation are different, but not clinically meaningful. Okay, so that is very important. So as a definition of biosimilar, there can be differences, but you have to show that they are not clinically meaningful. I'll show you here how they showed that this is not clinically meaningful. Okay, and this clinically meaningful part, you cannot do a clinical trial and show it's clinically not clinically meaningful, because that's a huge risk for the company, right? Because you are going to a clinical trial not knowing what the outcome would be. I mean, this is because it's a very costly trial and everything. So generally, this not clinically meaningful part you want to establish through your in vitro studies and biological activity studies in the lab itself, even before going to clinic. So if I go to the primary structure first. So this is, I discussed about all the methods and techniques that are generally used to understand the biosimilars and the biologics and show the biosimilarity. So this is how the output looks like, right? So this is, say, for example, primary structures. So say different say for example, this is light chain mass. Okay, so these are the mass spectrometry profiles, and as you see, they are trying to compare the profiles between rituximab China, your candidate biosimilar product, and the EU rituximab. And I mean, this is generally they show one representative figure, but this needs to be done for, as you see, that 10 batches of one product, 10 batches of the reference product and all. Okay. And then similarly, they have, this is amino acid sequencing or peptide map, as you see. So basically here they digest the protein with trypsin, where you get different trypsin cuts the protein at uh, K and R sites, and basically you get different peptides. And then by LCMS, you generally would able to get the identity of each of the peptides. So basically the whole sequence you can get. And also here as a profile, you want to match the profiles. So as you see over here, all the peaks here are matching, and if there are very even minute differences, you have to explain what these differences are coming from. Similarly, for the secondary structure or higher order structures, so most of the higher order structures are very qualitative analysis, where so this is, as you see, this is a DSC profile. So basically these are all matching, so basically all three are here, and this is the thermal melting temperature of the protein. So this basically shows that in a higher order structure manner, all these three products, the China rituximab, the biosimilar product, and the rituximab are similar. So similar for far UVCD or the near UVCD, showing the secondary structure or the tertiary structure, and even for IR spectra, which FTIR spectra, which shows the secondary structure. So basically, these are more qualitative assessment, where you try to match the profiles, or basically try to see all the profiles are overlapping with each other to make sure that these are similar in terms of higher order structure. Now coming to some numbers. Sorry. We talked about product related impurities, right? Aggregate or fragments, right? So here is a comparison of a size exclusion chromatography. So as you see, in general, if you run a size exclusion chromatography, I mean, you won't even see, because here we are talking about aggregates at a very low level, say maybe 0.2%, 0.3% level. So in general, you wouldn't be able to see anything, but if you zoom in, you'd be able to see this HMWS, higher molecular weight species or aggregates in there. So as you see over here, so these are all the aggregates. So it's very important to match the profile, in a sense that you want to make sure that there is no new aggregate or new species there in your product. So once it is exactly matching with the innovator product, so there is no risk of having a new variant or new impurity in your product. And then for an impurity, you always want to be in the lower side. So for something like HMW, you don't, even if you're not matching with the innovator. So as you see here, the percent aggregate in your, say, your Chinese rituximab is 1%, while EU it is 0.9%, wherein the biologics product is 0.4%. And these are the ranges. Right? Obviously, in this biologic product, it is low, but that is good, actually, because this is an impurity. So if you have a low impurity, that is fine. So you don't need to be matching the impurity if it is higher in the innovator product. And also, if when you put this product in stability, so when you store the product, right, so these impurities tend to increase. So it's always good to have a low impurity in the beginning, so that over the lifetime of the product, it can increase and go into these ranges. So these differences, fine. I mean, you really don't need to explain these differences, because there's an impurity and you are in the lower side. Okay. Similarly, for a fragment analysis, so this is a CSDS spectra. So where this big peak is the actual monomer or the actual rituximab, and these small peaks are different fragments. And as you see over here, if I try to see how much is that big peak present in there. So as you see in here, in this biosimilar product, it is more. So basically, this biosimilar product has less fragments than the sorry, this is the biosimilar product, has the less fragments than these two rituximab products. So which is again good. So although this range is, the monomer is higher, it is not matching the ranges of this rituximab, these two rituximab innovator products. We don't need to explain anything because this is again a purity, and basically it is showing this biosimilar product is more pure, little more pure than these two products. There's the innovator products. Okay, so now going to something where there are differences. So these are charge variant profiles. So I'm not going to into more details. So basically, when you run your product in a cation exchange chromatography or an anion exchange chromatography, it separates out based on the surface charge. Right now, this, as I mentioned, these biologics or biosimilar, both are very complex molecules, and it has many modifications into it, like oxidation, deamidation, glycosylation, everything. So that makes some part of the molecule more acidic or more basic, like that. So basically they separates out in a cation exchange chromatography. Okay, so that's what you see over here. So basically you see generally a big peak and some small peaks in a cation exchange chromatography, and whatever eludes before the main peak is called the acidic peak. This is AP, and after is basic peak, which is BP. So as you see here, you see here, even in the profile itself, you see this BP1 is much higher than this BP1 in the cetuximab, and that is also shown in here. As you see here, this is, sorry, this is 23% over here versus 10%, 10% in the rituximab. Okay, so this is a difference now. And also, if you see in the acidic variant, this is low, 11%, and this is 21%. So these differences there. So is this clinically meaningful? Okay, so one thing that they have already done is, so this MABs, this IgG monoclonal antibodies, always contain a lysine in the C terminal, which actually gets cleaved off when it is gets into the body, in the blood circulation. The carboxypeptide is B present in the blood circulation, cleaves that lysine off. Right now, what happens is when you make the make the protein, that lysine is still there, partly there. So basically, this, what they are showing is here. So they are treating this product with carboxypeptide is B and showing this BP1 is going down. So basically, they are showing this BP1 is actually C term because of C terminal lysine. Now, since the C terminal lysine anyway will get cleaved off in the body when it gets injected, so this doesn't make any clinically meaningful difference. So that's how it is explained. So here they have shown that once they are treated with CPB, that the carboxypeptide is B, the differences are less. And also, so they basically say that this difference is because of C-terminal lysine, which is not clinically meaningful. And that they have later shown in the biological assays. Okay. And one more thing that was not similar is the glycosylation. So as you see, these are the glycan profiles. So it's basically a complex carbohydrate chains that are at attached to the protein and attached to the FC part of the protein. And keep in mind, this product has a FC functionality, so it's very important. The glycan variants are very important for this protein. Okay, and this G0, G1, G2, these are different glycan variants. These are named based on the different chains, the different carbohydrates that are attached in the chain. Okay, so as you see over here, there are obviously some differences there. As you see, these peaks are much higher here compared to these, and also there are small peaks over here which are not there, so on and so forth. And also, if you go in terms of numbers, so see, this is called man is Manus. So this is the Manus basically. So it is higher in the biosimilar product and lower in the actual the innovator product. Similarly, for afucosylation, there are small differences are there, so on and so forth. So again, the question comes: these are some differences are observed. Obviously, you won't be able to make the exactly same biological product for a biosimilar. Now, are they clinically meaningful? So these are tested by the in vitro biological tests. So here I show the few biological tests that these are the outcome of the biological test. So I mean, just to have a flavor, just look into the these numbers only. So basically, as you see here, so they say they have analyzed multiple batches of China rituximab. So then the whatever percent potency they have got from say this FC gamma R1, 2, 3, these are different effector binding region of the FC of a product. And these are say like the EU rituximabs, and these are the biosimilar product. And this is the quality range they have designed from the mean plus minus 3 SD analysis of the innovator products. So basically what they show that, okay, and keep in mind these are in vitro biological tests, so these have high method variability. So as you see over here itself, so when we are testing different rituximab batches from China, so they are giving different numbers. They all are not 100%, right? So there is a spread always because of the method variability, because of the uh, complexity of the biological products. Okay, so that's why the range comes in here. So basically here they try to show that, okay, I have seen some differences in glycosylation. I have seen some differences in the charge variant profiles. But hey, here you see in the biological function, there is no differences. So most of the red triangles, which are the HLX product, are within these range. And few maybe out here and there, but they're like one batch and they're very close to the range. So basically, these biological analysis shows that although there are some differences in the physicochemical characterization, the biological functionalities are the same. Okay, so that way they have shown that the small differences that they have seen in the physicochemical characterization are not clinically meaningful. So this leads to the conclusion that taken the whole thing together, the stereosimilarity assessment demonstrate that it's a highly similar product, because they have shown everything like primary and higher order structure, post-translational modification, purity, impurity, and the biological activities and immunological properties. So these all taken together, this is called uh, this all taken together, they are saying that they are similar in terms of quality, efficacy, and safety, and that's how they are confirming that the this is biosimilar product. Now, after that, these are tested in non-clinical phase 1 and phase 3 trials, and it has been confirmed that they are biosimilar. But the base has come from the analysis that I have just shown, the analytical similarity assessment, where they have used multiple analytical techniques to establish the analytical similarity. Okay, so I had one more case study, but I will not go into it because it will take time. I'll rather go to the last slide. It's just a take home from the 1 hour 45 minutes I have talked. Is this biologics? Sorry, the spelling mistake. Biologics development, especially the biosimilar development. It's an emerging field of research, I would say. It's not only manufacturing and marketing and all. It's a huge field of research and definitely expected to grow further in coming years. And there are many well-established and startup biotech companies in India and abroad are investing in biosimilar business. And so for your students, MS or PhD students, so there are many opportunities in India and abroad in process and analytical sciences. So this is something probably I always say my faculty friends and even uh, not friends. I mean, I know faculties that just keep. I mean, ask your students to keep their eyes open for these. I mean, many a times these opportunities are not very well advertised, and I see uh, only students from metropolitan cities are getting into these positions. I don't see many are coming from the outskirts. So this is something probably I would request all of you to maybe ask your students to have a look around, be in LinkedIn, be in sites, try to get uh, connected to people in the industry. And most of these jobs are through, at least in India, I would say, through good connection. And yet there are some that are through walk-in interviews and all, but uh, a good portion of it through good connection. So making good connection, and to be frank, I got all my industrial jobs through LinkedIn connections. So it's a good way to get through, and there are many opportunities for sure. And for faculties, there are research opportunities in process analytical sciences, and uh, I mean, I know very few uh, faculty friends who are in this field, biosimilar field, mostly developing different processes or analytical sciences. And there is a huge possibility of collaboration with industry. So this is something, I don't know, is kind of missing in India, or maybe it's more from the industrial side that they want to have their confidentiality intact. But this is something some possibility that all of you can look into and explore to figure out new research opportunities for you as well as for your students. Thank you all for listening. Yeah, that's all from me.
Thank you so much, sir, for your valuable speech regarding the biologics and biosimilars. Now, from the audience, if you have any questions, you can ask, or you can write in the chat box also. And for you, the feedback link will be given to the chat box. To kindly please fill it.
Yeah, good evening, Dr. Avik Ghosh. Hello.
Hello. Yeah, please go ahead.
Yeah, good evening, Dr. Avik Ghosh. Yeah, myself, Durga Prasad speaking. Yeah, I have a basic doubt, like this biosimilarities or biosimilars. We are very new for the topic, and okay, as of now, we don't have a idea on biosimilars, right? You have given some basic requirement and how it can be done, everything you have given. But is it practically feasible, like in a organizations of like colleges? And
it's a good.
One more query, sir.
Yeah, go ahead.
Yeah, like what is the basic difference between bioanalytical method development and your biosimilar method development?
Okay. Yeah, so I'll take the first question first. Yeah, so obviously end-to-end development, I would not say would be possible in a university or even a bigger lab, even in IITs or ISCs. It won't be possible. The thing that can be done in a university lab is taking a smaller part, say like as I mentioned, the say clone development, right?
So that you cannot do the whole thing starting from clone development to actually the end product. Maybe the clone development part can be done in a biological lab. Similarly, for development of these methods, right? So there are many analytical techniques that are used. So, in say like for circular dichroism, so or for any methods, mass spectrometry based method. So these can be developed, or NMR, or any method. So these can be developed in the lab and then can be collaborated with the industries to use these methods in their use, basically. So basically, to in short, the end-to-end development is not possible, but you can pick and choose a few areas where it can be done in a lab environment. Does that answer your question?
Yes, sir. Fine. Right, sir. Thank you.
And can you just repeat the second one? Sorry.
Sir, second one is any analytical method development. Usually, when
Yeah,
we
yeah, sorry.
go for,
Big
HPLC,
difference
we call
between
it as
bioanalytical and this, right?
bioanalytical method development and validation.
Yeah, yeah. So there are, I mean, terminology wise. So generally, bioanalytical method development is termed the methods that you develop to actually analyze the pharmacokinetics, pharmacodynamics of the product. So basically, these are more in terms of when you come to the clinical trials. So you do, say, preclinical trial on animals or PKPD studies on healthy volunteers, right? So after the study is done, you get the blood samples. Then you should have methods to see the how the pharmacokinetics is, how the pharmacodynamics is. So those methods are generally termed as bioanalytical method. So these are used for biosimilar development. So generally, in all the companies, there are teams who develop these bioanalytical methods and analyze these samples, or actually I'd say 50% of companies outsource this to CROs who does it. So there are guidelines, specific guidelines on how to develop a bioanalytical method and validate the bioanalytical method. So I have roughly used qualification and validation in same meaning in my presentation, but they are a little different. So qualification is more a lighter version of a validation, I would say. The validation is more stringent version. And so these, if you're interested, there are FDA guidance and EMA guidance how to do bioanalytical method development and qualification. So I can share with you, or you can. These are easily found in the net also. You can go through it.
Sir, one more a small query.
Yeah.
In case of bioanalytical method development, when it is being done, we need to take permissions of the ethics committee. Either it may be
Yes.
animals or on humans. So in the
Yes.
same case, in biosimilars also, is it the same case?
Yes, yes. So I mentioned about this clinical trial application in the pathway. Yeah. So here you say this clinical trial application. So this basically has two parts. One part is the regulators go through that our dossier that we file. They review it in terms of how we are showing the analytic biosimilarity and all. And also we need to provide your clinical trial bioanalytical methods and everything over there. So that needs to go through the ethical committee approval of that particular health authority that we are targeting. So if we are doing the clinical trial in, say, Hungary or any other country in Europe or in India or in Japan, so it needs to go through the ethical committee. Any trial, it may be a animal. So basically, most of the developed countries in, say, like for FDA or EMA, the animal studies are not permitted, in a sense that they say that these are not very relevant. So you don't need to do that. But wherever animal studies needs to be done, it needs to go through the ethical committee approval, as well as for a healthy volunteer or even for the clinical studies. So this ethical committee approval is part of this clinical trial application.
Thank you, sir. Yes.
Thank you.
Any more questions from the audience? And for your kind information, the feedback link has been shared in the chat box. You can click it.
Sure.
Okay, thank you so much. I think no more questions from there.
Yeah,
from the
thank you, and
audience.
thank you for having me here for the presentation, and thank you.
Thank you so much, sir, for giving me us the valuable speech regarding the biologics and biosimilars. And thank you once again all the audience who have joined this online activity program. Thank you so much.
Thank you. Have a good day.